Hey everyone, welcome to Piazza Research, and we are continuing the human physiology illustration, exploration, concept map overview of how the human body works. And you can get a free copy at my website, piazzaresearch.org, to follow along and reference for yourself. Also, if you haven't already, consider checking out my book on psychiatry or neurology. Neurology is coming very, very soon here. It's really good. I put a lot of work into it. It's very exceptional. A lot of brain imaging, really clear illustrations and graphs, as well as concise wording that allows you to actually remember the content. So consider checking that out. And with that, let's jump right back into the nervous system. And its focus will be on the neuromuscular junction. Now you can see here, I drew a lower motor neuron moving along the clavicle and into the upper arm, maybe the deltoid muscle it's innervating, and I expanded that out, and we can see how all of that works. But first, I just want to mention that neurons can be upper or lower motor neurons. Upper just means it originates in the brain and moves down into the spinal cord, whereas a lower motor neuron originates in the spinal cord and branches out into the periphery of the body, part of the peripheral nervous system. So let's say the nerve we're following and looking at entered the deltoid muscle and is innervating it. As you can see here, I drew a neuron as well as a muscle. Now muscles and their structure can be a bit confusing, a lot like the movie Inception, where you had a dream within a dream within a dream, or maybe some will say nightmare. And muscles are kind of structured in a similar way, where you have matter, cylinder-shaped matter within cylinders within cylinders within cylinders. And so it can be a bit confusing to organize your thought process and where things are situated. But I think I've done a good job of illustrating exactly what is going on. So the very first macro-sized part of the muscle is the muscle bundle. You can see here I drew nerves innervating this muscle bundle. There are arteries and veins within there providing blood supply. And you have calcium channels throughout. And the muscle bundle is made up of a smaller cylinder-type structure called the fascicle. The fascicle itself is made up of muscle fiber or muscle cells. And so I've gone ahead and drawn a muscle cell here, scaled up so we can see what's going on. And then before we get into the physiology of what's going on that allows muscle contraction to take place, why don't we briefly look at the structure of a neuron, in this case a lower motor neuron. This guy has dendrites, which is the receiving end of a neuron typically, receiving any signaling. Then there's the soma body of the neuron, which encases the nucleus. And you have the axon hillock, which is typically the origin story of an action potential. That's where a stimulus will trigger your signal to begin. And we can look into a little bit more detail of how that occurs. So we're talking about an action potential firing, what this really means, and how we discuss it and illustrate it. What we have here is on this graph right here on the side, we have a neuron. The interior is in between the two blue lines. The exterior is outside it. Initially, minus signs represent a negative membrane potential relative to the outside. And this is typically going to be its resting potential. It's going to be at a level of negative 70 millivolts to maintain a resting potential. You don't want your neurons constantly firing uncontrollably, where you have a positive membrane potential, or if it's depolarized, because then this could be causing serious symptoms that we see in neurological deficits and disorders. Constant muscle contractions or twitching, which is uncomfortable and problematic. So the body has found a way to kind of maintain this resting membrane potential of negative 70 millivolts by allowing potassium and sodium to actively move in and out through either passive leaking and passive transport or through the sodium-potassium ATPase pump. This allows the membrane potential to either move towards negative 70. If it's getting too negative or too positive, move back to negative 70. Once an action potential fires, sometimes we represent it by these plus and negative signs, and the inside would be positive if an action potential is firing. If the axon's radius or diameter is increased, the action potential propagation also increases. This is the opposite of fluid, where if you increase the diameter of a hose, for example, the fluid rate or speed or velocity will actually decrease. Whereas if you narrow the cylinder, the fluid rate increases. So a nerve signal is the opposite, and action potential is the opposite. So you might feel some sort of stimulus or trigger, and it may not be enough to reach the threshold of negative 55 millivolts, and your action potential doesn't fire, and no muscle contraction can take place. If the stimulus is enough, where it reaches the threshold, moves way up, the neuron depolarizes. This triggers the opening of a potassium channel. This allows potassium to move out, again moving that millivolt back to a negative level, in this case negative 70. It'll go past it briefly. It's called hyperpolarization, and then it once again will reach its resting or equilibrium point at negative 70 millivolts until another stimulus triggers it. That is how a healthy action potential operates. So that signal moves along the axon and into the motor neuron terminal, which I've drawn here. And in here we have the mitochondria. We have our beloved neurotransmitter acetylcholine hanging out in a vesicle. And the calcium will move in, allowing this action potential to continue propagating. Acetylcholine will then bind to its receptor. There are two receptors it binds to. In this case, it's binding to a nicotinic acetylcholine receptor, NACHR, which is prevalent within the neuromuscular junction. The other receptor it could bind to is muscarinic receptors. These are receptors that activate the parasympathetic nervous system, but they aren't typically found in the neuromuscular junction. And so, once it binds to this various different subunits of the receptor, sodium is able to flow in, again allowing this action potential to continue operating and propagating through what is called the sarcolemma. I think I'm pronouncing that right. And into a T tubule. Once in the T tubule, calcium can once again enter, this case, a muscle cell and allow for the contraction to actually occur. The contraction operates within another cylinder within the muscle cell called the myofibril. The myofibril is made up of those cool band-looking patterns you might have seen before called sarcomeres. We are going to briefly break down exactly what is happening with the sarcomeres once that calcium enters and allows contraction to occur. What we are following is called the sliding filament theory. The sliding filament theory is what is proposed as the best illustration of what is happening. And so we have a structure called actin and myosin. Myosin, we'll say, is represented by the blue here and here. Actin is these white circles. Actin is wrapped by tropomyosin, and on this tropomyosin is something called troponin. Calcium is able to bind this troponin. Once it does so, the myosin head, the blue part, is able to bond with the tropomyosin. ADP and phosphate is attached to the myosin head. It moves off once the myosin head attaches to the tropomyosin. After it leaves, this is called the power stroke. Literally, the myosin head changes its position in 3D space. We call it a conformation. This movement from like a greater than 90-degree angle to a 90-degree angle allows these actin to move closer together. So previously, the actin, this white, they were spaced out. Once the blue part performs the power stroke, the actin, the white part, move closer together, and the sarcomere becomes shorter in length, thus being a contraction. After the power stroke takes place, the contraction takes place, ATP will once again bind to the myosin head, and the myosin head will once again undergo a conformation change back to its original position, and everything starts over as it was before. And this happens over and over and over and over again. Some important terms to remember and easily remember when looking at these band patterns. We have M. You can think of it as middle of myosin, so it's the middle of the blue part. We have I. I is a thin letter, and so you can think of actin, which is also thin. That's the white part. You have H. H is thicker than I. It's basically two I's with a line in between them. This is the myosin, or the H line. And then the final line to remember, and there's more, but these are the most important and crucial ones. The Z line, you can think of Z, the end of the alphabet, is the end side of a sarcomere. So that is a good rundown of the neuromuscular junction, and I think we'll wrap up there, part 2 of the nervous system. And I'll see you guys in the next one. Take care.